This Embryology Highlights screencast mentions flexures and cardiovascular development and then focuses on placentation and fetal membranes. The early embryo is C-shaped because it has flexures in the head and the tail region. The cardiovascular system develops early in the embryo as the embryo enlarges to the extent that diffusion becomes insufficient for waste removal and delivery of nutrients. Blood vessels begin as blood islands in splanchnic mesoderm of the yolk sac and the allantois, and these blood islands arise by mesenchyme cells forming red blood cells and then inducing the surrounding mesenchyme to form endothelium. The individual blood islands then sprout buds and they coalesce to form vessels. This image of a pig embryo shows a flexure in the midbrain region, a flexure in the cervical region, and flexures in the tail region to give it this C shape. In our domestic mammals, these flexures largely straighten out so that the head ends up being aligned as an extension of the vertebral column, unlike primates where the head faces forward and perpendicular to the vertebral column. This image of an early embryo with the somites shown in orange and the digestive tract shown in yellow and the allantois growing out shows the cardiovascular system with veins shown blue and arteries shown in red. Early in the embryo, the vessels are paired. There is bilateral ventral aortae, bilateral dorsal aortae. The ventral aortae fuse to form a heart, and the dorsal aortae fuse to form the aorta as seen in the adult. Bilaterally, the ventral and dorsal aortae are joined by aortic arches, which run in the pharyngeal arches. Venous blood comes from both the embryo itself via cranial cardinal vein and a caudal cardinal vein, and also from fetal membranes. So the umbilical vein carries blood from the placenta, and early in the embryo, the Vitelline veins are carrying blood from the yolk sac. The allantois and the yolk sac are also, of course, going to receive arteries, which are not shown in this image. The placenta represents regions where the uterine wall and fetal membranes are in contact with one another so that metabolites can be exchanged and waste can be removed throughout the pregnancy. There are different placental types based on the contact areas. For example, the pig has a diffuse placenta, whereas primates have a discoid placenta, carnivores have a zonary placenta, and then ruminants have a placentome, as does the horse. In addition to typing based on the areas of contact, placentae can also be classified based on the histological layers that exist between uterine blood and the fetal blood. This shows types of placentae. The dark areas represent areas of functional contact, and the brown, light brown areas represent other areas of chorion. So the pig has a diffuse placental contact area. Virtually the entire placental surface is involved in exchange. In contrast, the ruminant has discrete areas of functional contact which are called placentomes. There are 4 rows of placentomes. The equine also has discrete placentomes, but they're very small and distributed throughout the surface, and so these are referred to as a microplacentone, or what we're looking at are microcotyledons, the cotyledon being the fetal part of the placentome. Here, these are all distinct cotyledons. In the case of the human, the area of functional contact is shaped like a disc, and so this is a discoidal placenta. Rodents also have this kind of placenta. In carnivores, the dog and cat, functional placenta represents a band that goes around the center of the conceptus, and so this is called a zonary type of placenta. Polar ends are plain chorion, which is not functioning as a placenta, and then the zone of functional placenta is bounded bilaterally by these marginal hematomas, which don't have any placental significance. This cartoon shows a placentome of a ewe on the left and a cow on the right. In green, then, is the cotyledon, which is the fetal structure composed of chorion and allantois deep to it. And the uterine wall is in orange, and the uterine epithelium is in yellow. And so you can see that fetal and uterine tissues interdigitate for very close contact and exchange of nutrients. The ewe is different from the cow. Cow has a convex coracle, and the ewe has a concave coracle. So the maternal component of a placentome is a coracle. The fetal component of a placentome, shown in green here, is a cotyledon. This image is of a dog placenta, a zonary placenta of a dog. It was actually a cadaver dog. On the left here, the uterine wall has been incised and the muscle has been pulled away, but the uterine lining is left in place. We can see a zone here of uterine mucosa. We can see through that marginal hematomas and the rest of the conceptus. In this middle image, the uterine lining has also been reflected. And so now we're looking at fetal membranes. And we have the zonary placenta bounded by marginal hematomas. And then this is just the chorionic surface at each polar end, which is not involved in the functional placenta. In this last image, what we have done is incised through fetal membranes and reflected them. And so now we're looking at the inside of the placenta and the marginal hematomas. And we see vessels coming from that to a fetus, which is located right here. Considering next fetal membranes, each conceptus develops 4 fetal membranes. Chorion and amnion develops the allantois and the yolk sac. This image shows the development of the 4 fetal membranes. In blue here we have the fetal membranes that develop from the trophoblast from somatopleur, and they are the chorion that forms this entire outer surface, and then the amnion here, which is formed by folds of chorion that meet and blend together, creating an amnion. Shown in yellow are the 2 fetal membranes that develop from splanchnopleur. They are, first of all, the yolk sac, which developed from the original hypoblast layer, connected then to the gut when endoderm was formed. Growing out of the hindgut is a allantoic cavity, or the allantois. This image shows a drawing of pig fetal membranes with an emphasis on the mesoderm, shown in red. The chorion, and you can see the amnion developing here. The mesoderm associated with those is somatic mesoderm, whereas the yolk sac and the allantois, which is growing out here, mesoderm that surrounds those is splanchnic mesoderm. The significance is that only splanchnic mesoderm has vessels, so only the splanchnic mesoderm then can form a functional placenta. Since the yolk sac soon atrophies, it is up to the allantois to carry the vessels out, make contact with the chorion, which remains at the surface. As these 2 mesoderms merge together, we now have a vascular area of fetal membranes that can participate in a placenta. This image shows the fetal membranes colored. You can see that the yolk sac is regressing and the allantoic cavity is expanding. The amniotic cavity is shown in blue. Chorionic cavity is shown in brown. What's going to happen then is the allantoic cavity is going to go out and make contact with the chorion. This colored drawing shows 3 stages of fetal membrane development in the pig. Chorion lumen is shown in tan, the amnion in blue, and the yolk sac in yellow, and the allantois in green throughout these images. This is an early stage of development. The amniotic cavity is just being formed by folds from the chorion, because the yolk sac, which is continuous with the yellow gut here, is very large and can actually be a functional placenta for a short time in some species. Meanwhile, the allantois is just beginning to grow out. When we get to a later stage of development, here we see the yolk sac is regressing and the allantois is growing out so it can make contact with the chorion and be a functional placenta. The amnion is separated from the chorion as these two folds have merged together. This later stage of development shows that since the pig has a diffuse placenta, the allantois has grown out to make extensive contact with the chorion, carrying vessels to it. The tips of the chorion are sometimes referred to as necrotic tips in those regions that don't get a blood supply. Allantois has grown around and grown between the chorion and the amnion, which is shown in blue here. The pig can be born with the amnion around it, while the allantois and the chorion remain back as afterbirth. This image compares the chick to the pig. In the bird, of course, the yolk is going to be the main source of nutrient, and so the yolk sac persists, is going to be very large. The allantois does develop. In the pig, the yolk sac is going to regress and the allantois is going to grow out and become very large. We can also compare the pig to the human. In the human, there's a discoidal placenta. The allantois carries vessels out to that region, but there is no significant allantoic cavity in contrast to the pig, where the allantoic cavity is large because the allantois has to make extensive contact with the chorion, since this is a diffuse kind of a placenta, in contrast to the human. This last image is intended to show another difference between the human and our domestic mammals. In the human, the conceptus grows within the wall of the uterus, whereas in our domestic mammals it grows within the lumen. Here we see 3 different stages of development in the human pregnancy, and the conceptus is growing in the wall. That concludes this Embryology Highlights screencast dealing with flexures, cardiovascular system, placentation, and fetal membrane.